So we already know that chromosomes are made up of really long strands of DNA all wound up into themselves. So something like, well, I'm just kind of drawing it as a random long strand of DNA all wound up in itself. And on that strand, you have sequences which we call genes. So that might be one gene right over there. This might be another gene. That might be a gene right over there. And each of those genes can code for specific polypeptides or specific proteins. And the key question is how do you go from the information encoded in these genes, encoded as sequences of DNA, how do you go from that? How do you go from the gene, which is encoded in DNA, how do you go from that to protein, which is made up of polypeptides, which are made up of amino acids? And this is often called the central dogma of biology. But we already saw in the video of transcription that the first step is to go from the gene to messenger RNA. That the RNA, the messenger RNA, you can view it as a transcript. We have rewritten the information now as RNA. And then the next step, which we were going to dive into in this video, is going from that messenger RNA to protein. And this process is called translation, because we're literally translating that information into a polypeptide sequence. And you can see a little bit visually here, and this is all review. We covered a lot of this in the video on transcription, and the overview video on transcription and translation is if you look at a eukaryotic cell and the bacteria in a prokaryotic cell, it's analogous. You just don't have the nuclear membrane, and you're not going to do the processing step that I'm going to talk about in a little bit, and we went in detail on the video on transcription. But you start with the DNA. You have your RNA polymerase as the main actor that's able to transcribe the RNA from that. If we're talking about a eukaryotic cell, what you end up with, we wouldn't call mRNA, we would call that as pre-mRNA. Pre-mRNA, which then needs to be processed. The introns need to be taken out. We add a cap and a tail here, and if we're talking about a eukaryotic cell, we'll then formally call that mRNA. And then it can travel, and this is where we get into the translation step, it can travel to a ribosome, which is where it will be translated into a polypeptide sequence. And you see the analogous thing happening here in this bacterial or this prokaryotic cell right over here, except you don't see the nuclear membrane because this is prokaryotic, and you don't see that processing step. So you could just consider this straight. This is mRNA right over there. So the questions are, well, how does this thing happen, and what even is a ribosome? So let's zoom in a little bit on a ribosome right over here. And there's a couple of interesting actors. One, as you can imagine, is the ribosome itself, and it is made up of proteins plus ribosomal RNA. So in the video on transcription, we're already familiar with messenger RNA, and we often view RNA like DNA as primarily encoding information. It's acting as a transcript for a gene, but it doesn't have to only encode information. It can also, so it's proteins plus, that's not a T there, this is a plus. It can also provide a functional structural role, which it does in ribosomal RNA. And this big, this looks like an oversized hamburger bun or something right over here. This is a super oversimplification of what a ribosome looks like. And I encourage you to do a web search for image searches for ribosomes, and then you can get a more appreciation of how beautiful these structures are and how intricate they actually are. So this is the site, and you can broadly think of the ribosome as having this, this is the top bun and the bottom bun, and it's going to travel along the mRNA from the 5' end to the 3' end, reading it and taking that information and turning it into a sequence of amino acids. So how does that actually happen? Well, each of these every three nucleotides there, we call that a codon. So that's a codon. This is, let me do this in a color that is visible on both white and black. So this next 3 nucleotides is a codon. This is a codon. This is a codon. And what's actually, the information is actually encoded in the nitrogenous bases. So this first codon right over here, we see it's AUG, so the nitrogenous bases are adenine, uracil, and guanine. And this has this codon, it codes for the amino acid methionine, but this is a good one to know. AUG, let me write it over here. AUG is known as the start codon. Start codon. This is where the ribosome will initially attach to start translating that messenger RNA. And so the way that this drawing is that we are just starting to translate this messenger RNA. So how does that actually happen? How do we get from these 3-letter sequences to specific amino acids? Well, let's think about it. How many possible 3-letter sequences are there? Well, there are 4 possible nitrogenous bases there. So there's 4 possible. So if you have a codon and it has three places, there's 4 possible things that could be in the first place. There's 4 possible things that could be in the second place, and there's 4 possible things that could be in the third place. So there are 64 possible permutations. 4 times 4 times 4 permutations. So you could think of it, there's 64 different codons, different ways of arranging the A, the U, and the G. And that's good because there are many amino acids. And this is actually overkill because there's actually 22 standard amino acids, 22 standard amino acids, and 21 that are typically found in eukaryotic cells. So we have more than enough permutations to cover the different amino acids. And it's not hard to find tables that will actually show us what the different sequences, what they actually code for. So you can see here, you can take the first letter, the second letter, and then the third letter. Look at the different sequences, and you can say, okay, look at that. AUG, adenine, uracil, guanine, that codes for methionine, right over here. You could do that with any of them. You could say cytosine, uracil, that codes for leucine. And you can see that it's not just one amino acid per codon, that here you have 4 codons, all code for leucine. And so it turns out that 61 of the codons, let me write this down. So 61 of the codons, of the possible 64, code for amino acids, and 3 play a role that essentially tells the ribosome to stop. 3 codons are stop codons, and you can see them right over here. UAA, UAG, UGA. That's how the ribosome knows to stop translating. So AUG, that's a start codon, and it codes for methionine. So that lets you know that, well, these polypeptide chains are going to start with methionine, and then these characters tell it where to stop. But how does the amino acid actually how do they all get tied up together to form this polypeptide? And how do they get matched up? How do they actually get matched up with the appropriate codon? And that's where we have another RNA-based actor, and this is tRNA. So tRNA, the T stands for transfer RNA. There's a bunch of different tRNAs that each can bind to specific amino acids, and on parts of those tRNA, they have what are called anticodons that pair with the appropriate codon. So this tRNA, and that's not what it looks like. I'll show you in a second what it looks like. That's a tRNA molecule, tRNA. At one end of the molecule, it's binding to the appropriate amino acid, methionine, right over here. And then at the other end of the molecule, although it's in the middle of the tRNA actual chain, you have your anticodon. And your anticodon matches up to the appropriate codon. And so this is how, if they bump into each other the right way and the ribosome is going to facilitate it, that the AUG is going to be associated with the methionine. And if we look at what tRNA actually looks like, and this is still just a visualization, so this is a strand of tRNA. You get a sense of, okay, it's a sequence of RNA right over here. This is I guess you could think of it, it's 2-dimensional structure, but then it wraps around itself to form this fairly complex molecule. and the anticodon, which is right here, it's kind of in the middle of the sequence, it forms the basis for this end of the molecule. That's the part that's going to pair with the codon on the mRNA. And then at the other end of the molecule, is where you actually bind to the appropriate amino acid. So I know what you're thinking. All right, I see that the ribosome, it knows where to start. It starts at the start codon. I see how the appropriate tRNA can bring the appropriate amino acid. But how does the chain actually form? And you can view this in 3 steps, and associated with those 3 steps are 3 sites on the ribosome. And the 3 sites, we call this the A site. You're not going to be able to see it if I write it in black. A, or yellow. All right, let me write it in blue. So that is the A site. This is the P site. And this is the E site. And I'll talk in a second why we call them A, P, and E. So the A site is where the appropriate tRNA initially bounds, the tRNA that's bound to an amino acid. And so you can see we're starting the translation process. The next thing that's going to happen is another tRNA, the one that has an anticodon that matches the UAU, that's going to bond over here on the A site. And it's bringing the appropriate amino acid with it. It's bringing the tyrosine with it. So why is that called the A site? Well, A stands for aminoacyl. You can, or an easy way to remember it, it's the tRNA. It's the place where the tRNA that's bound to an amino acid, just one amino acid, is going to bind on the ribosome. And so once that happens, once this character comes here, let me draw that. Once this character comes right over here, there's going to be AUA, and it's bound to the tyrosine. Well, then you can have a peptide bond form between the 2 amino acids, and the ribosome itself can move to the right. So this tRNA will then be in the E site. This tRNA will then be in the P site, and then the A site will be open for another amino acid carrying tRNA. So what does P and E what do the P and E sites stand for? Well, you can see a little bit more clearly right over here. So the P site is where you have the polypeptide chain actually forming. And so the P site is often, one way to remember it is that's where you have the polypeptide chain. And now you have a new A site where you can bring in a new amino acid. And then the ribosome is going to shift, once this is bound, the ribosome, the peptide bond forms, and then the ribosome can shift to the right. When the ribosome shifts to the right, we're going to be in this position where the thing that was here, that was in the A site, now the polypeptide is attached to it. It is going to now be in the P site, and the thing that was in the P site is now going to be in the E site. It is now ready to exit, and that's why it's called the E site, because that's the site from which you exit. And so this is going to keep happening until we get to one of the stop codons. And when you get to one of the stop codons, then the appropriate polypeptide is going to be released, and we will have created this thing that could either be a protein or part of a protein. So this is very exciting because this is happening in your cells as we speak. And in fact, if you think about things like antibiotics, the way that they work the way that antibiotics work is that ribosomes in prokaryotes are different enough than ribosomes in plants and animals or in eukaryotes that we can find molecules that hurt the function of ribosomes in prokaryotes but don't do it to eukaryotes. And so if you have bacteria in your bloodstream and if you take the appropriate antibiotic, it could disrupt this translation process in the bacteria but not in your cells that you want to keep.